Oh my gosh.
Van Raden.
Van Raden. He said, Dr. Wall just said he wanted to sit in the audience so he could see Dr. Van Raden's slides. So if a guy like Dr. Wall wants to see your slides, that's high praise, I figure. Are we ready? There's a thumb up in the back. Okay. Dr. Paul Van Raden earned his Bachelor of Science from the University of Illinois and his PhD from Iowa State University. He has a dizzying array of publications, many of them in the peer-reviewed Journal of Dairy Science. He studied interbreeding, crossbreeding, the impact of selecting brown Swiss, Jersey, and Holstein bulls. He knows about embryo defects, stillborns, and calving ease, which everyone knows the cows will be happy to hear about. Dr. Van Raden studies early lactation and traits in mastitis, a painful infection of the udder, and extended lactation. He's an expert on how predictable treatment impacts value and economic merit. He is also involved in inter-bull annual meeting proceedings, and I wanted to know how inter-bull proceedings were different than intra-bull or just plain bull proceedings. Currently, Dr. Van Raden is a research geneticist working for the Animal Improvement Program of the USDA. He'll be speaking about 29 minutes. I welcome you to give me your cards, and he'll be followed by Dr. Wall. Thank you.
Thank you very much, Barbara, for the nice introduction, and thanks for inviting me here. This is really my first chance to try to explain this topic to an audience that's not 100% agricultural. So hopefully you'll be able to understand the ideas even if I can't speak as clearly as a lawyer would. Okay, so animals have improved slowly over the thousands of years since they were first domesticated. And now we try to make each generation better by choosing from the best genes available from the previous generation. Let's see. Okay, so when I was younger, I actually went out and inspected animals as a job. Now that job is done by many other people, and we receive in my laboratory the data from about half of all the dairy farms in the US, and we also receive data from 25 other countries. And our job is to analyze that data, to give advice to farmers on what genes to put in the next generation. And specifically, we tell them, we give them a list of bulls available to choose as sires of their calves. That's the main business. And then we also have the same for females to choose. Okay, so what are the main goals of animal breeders? The one that I'm most concerned with is to provide the world's food needs, the standard traditional agriculture. But we can think of adapting our animals just as we have to learn to adapt and do new jobs as time moves on. Animals may need to do or they could do new, more profitable jobs. Some of these, such as there's companies that breed guide dogs, and they tried to have a genetic selection program to make them most effective. Of course, draft horses were developed, but their jobs were mostly taken over by tractors. If we run out of fuel, maybe they'll make a comeback. A lot of animals provide companionship or entertainment, and of course, you would not want to have a wolf in your house, but a puppy and a dog is a nice companion. And there's also the new end with possible pharmaceutical production. In any of these cases, the progress is faster if we use modern selection tools, and that's what I'll talk about. Okay, but first to review the tools that were used in nature by Mother Nature to develop the animals that we started out with. And these tools that Mother Nature uses are the well-known forces of starvation, freezing to death, injury, infection, disease. Many animals were eaten by predators. The selection process for males is that they would, in a lot of species, battle each other in order to have the right to mate. And in all populations, harmful mutations gradually occur over time, and those are sort of weeded out by embryos aborting and young animals dying from harmful mutations. So Mother Nature does obey the law. It just happens to be the law of the jungle. And I think we can do better than that with our artificial selection. So how do the tools that we use compare? One tool that we really want to focus on is we try to specialize these populations. It's not really speciation, but just making them specialized. And we do that by using a fence or a barn or a house so that you have control over the mating population instead of letting them randomly mate with whatever other animals there happen to be. After we've improved these genetic lines, they can be easily imported or exported to basically anywhere on Earth. The tools we use in the dairy industry, primarily artificial insemination and progeny testing. That's where we get a sample of offspring first to determine if that bull has a good set of genes, and then after the initial test, then we'll use that bull much more, produce many more offspring. And the embryo transfer is basically the same tool, but for females, is that instead of having an elite cow have many calves, the embryos are transferred into surrogate mothers so that her genes can be propagated much more often. And another tool more recently developed is marker-assisted selection, where we can actually trace the genes that have been inherited directly using DNA tests. And animal breeders generally obey economic laws. Will use the tools if they make a profit, and if they're not profitable, they don't get used. Some of the reproductive tools that have been developed, just a quick history. In 1953, a company figured out how to freeze semen. This is on the left is a picture of the first calf born as a result of frozen semen. And within only about 2 years after that, there were hundreds of thousands of calves being born with this technology. And today, about 80% of all the calves born result from frozen semen. Then embryo transfer, the middle picture is the adult bull, but he was the first embryo transferred from one cow into another. And this technology caught on immediately. This bull himself had 6,000 daughters. So you can see that the farmers were not afraid of this technology. There was no law for or against it, and it just took off. And now there's 25,000 calves per year born by this. Embryo splitting on the right side, you see these two bulls here. They were an embryo that was divided in half under the microscope, and the two halves implanted and resulted in those two bulls. Once again, for the two bulls, we had 600 daughters of this one and 500 daughters of the other one. And based on that data, we did confirm that the traits of the daughters were basically identical. And then some potential future tools is the ability to sort the X and the Y bearing sperm. That's really catching on now. Embryo splitting, as I just said, there is some use, but it really hasn't caught on just because the economics. It's too expensive to do widespread use of it. Adult cloning and transgenics, I'll let Bob Wall, the next speaker, he's the expert on that, and you'll hear more about those from him. A topic that I'm very much involved in is genomic selection. That's where we have the ability to precisely determine what genes were inherited by say a calf, so that you can do the selection early in life instead of waiting to measure the traits that are expressed later. Okay, as a result of these technologies, we're able to get very large family sizes and make this whole technology pay off. And the progeny testing, usually we obtain about 100 daughters as a first test of the bull's genetic quality. Measure the important traits on his daughters and then select the best bulls to be used further. And as an example of that, this is sort of an extreme case, is this one bull, Elevation. He was born in Virginia and lived the rest of his life in Ohio. He had more than 80,000 daughters, 2.3 million granddaughters, and 6.5 million great-granddaughters. So clearly that once we identify a bull we like, we do in fact make good use of them. The traits that are evaluated, my laboratory has been doing these bull rankings or bull evaluations for more than 70 years continuously. The two traits they started out with were milk and fat production of the cows back in 1935. Then we switched to mostly emphasizing protein, the protein content of the milk since 1977. Calving ease evaluations began in 78. The udder measurements of the udder shape and the udder height off of the ground in 1983. Feet and leg traits, whether the cows could walk properly, the size of the cows, longevity, mastitis resistance, fertility, and the rate of stillbirth. So whether the calves are all born healthy and alive, that's a trait that we just added last year. Then this shows that when you have all those traits, somehow you have to decide which ones are more important. And these are how we emphasize the traits. The protein and the fat yield, because to make cheese, that's the main use of our milk now, is to go into cheese making. It requires both protein and fat. But we now put a lot of emphasis on the longevity of the cows, mastitis resistance and fertility, the shape of the udder also receives emphasis because that allows the cows to be milked more easily. Calving ease and stillbirth, we also select for those, but we can't make quite as much progress for those traits, so we don't put quite as much emphasis on it. It's really, according to economics, is how we derive these weights. The data analysis then, we get data from, we have 200 million data values that we use to compute the rankings. Use a relationship matrix among all these bulls and cows. And when we get done calculating the US rankings, we send the data, our data file on to Sweden, and they combine it with, this is the inter-bull portion of the talk. And in Sweden, they combine our data with data from 26 other countries, and then we have a global ranking of who the best bulls are. I think Sweden was chosen because they were viewed as a neutral country. Okay, so when we get that file back and we look who is the best bull at the moment, and conveniently his name happens to be Justice. I didn't make this up. And last year from Justice, the semen sales were 198,000 units. That means 198,000 cows out there were inseminated with frozen semen from this bull. The price listed was $40 a unit. If you multiply that out and over the lifetime of the bull so far, he's made about $30 million of income for the company that owns him. And in our data file, we have 12,000 daughters with milk production records, 10,000 of those in the US. He also has about 600 daughters in France, 500 in Italy, 400 in Denmark, 300 in Netherlands, and about 4 or 5 other countries. And soon they will be all over the earth. Now, why do people care so much to invest in this one bull? Because if we look at the traits of his daughters, this is a picture of one of his best daughters, but we report the averages. The average cows in the herd would give about 10 gallons a day. His daughters give 10.5 gallons a day. The protein production is quite a bit higher for his daughters than the average cows. The cell count, which is our measure of mastitis infection or health status of the udder, his daughters are much healthier. They live more than 6 months longer than average. The fertility is much better, and the trait, the calving difficulty, on the average, 8% of the calves are born with difficulty, but for his calves, only 3%. That's a huge difference, and that's why the farmers want to use this particular bull. Okay, so the mating is not at random, as it sometimes is in the dormitories and sororities. The mates are chosen by computer programs for about half the cows in the US. The pedigree of the bull and cow are compared to try to minimize inbreeding. Carriers of the same genetic defects are not mated together. Weak traits of the cow are matched to strong traits of the bull to try to optimize the offspring. And if it's the first calf that a cow is going to have, then we'll use one of these bulls like this Justice that has very easy birth, because the first birth is the more traumatic. After that, they're a piece of cake, right? So have we made any progress? In 1957, or 50 years ago, cows were milking about 2.5 gallons per day. Now the average is up there close to 7.5, so cows are giving 3 times more milk than they did 50 years ago. Is progress possible for other traits? Well, there are some single genes that have very large effects. This is a picture of a Texas Longhorn. In that breed, of course, all of the cattle, all of the members of that breed have horns. That's just one of their properties. But there are other breeds, such as the Angus, black Angus, that 100% of them don't have horns. And it's just a 1 gene difference. And that gene is dominant. So if you bred this cow to an Angus bull, all the calves would not have any horns at all. So that's, we can change the animals very quickly. This is a naturally occurring gene. It didn't require any manipulation. And in our Holsteins, the dairy cattle, we really haven't taken much advantage of this yet, because other genes that we're selecting for are more valuable. But there is the opportunity to change the population quickly. The specialization occurs very much in cattle. We have dairy breeds and beef breeds, and beef are not selected for milk traits because they're not milked, and dairy cattle are not selected for beef traits because the focus is just on the milk production. In chickens, you have the same thing. You have chicken varieties that are selected only for meat yield or only for egg yield. And once those improved varieties are created, they are marketed and distributed all over the whole planet. The breeding companies themselves in the poultry and swine business, they tend to have closed breeding populations where the company will maintain its own population, and they tend to have central control because of the brand name process that the farmers basically have to use the varieties that Frank Perdue or Tyson or whoever, the Smithfield, tells them, these are the genes that you're going to use for our product. Whereas in dairy and beef, we have more of an open exchange of breeding stock. The producers choose what genes they will use based on these genetic rankings calculated by us or by the breed societies. And until now, we've had very little patenting or intellectual property, but that may change because as we go into this tracing individual genes, there's a, should that process be patented or not is one of the questions that comes up. Then, just started to see how we're doing in terms of my main job, which is to feed people. Is the price of animal protein, in this case, competitive with that from plants? Because as we improve our animals, there's also crop breeders that are constantly improving the crops, and the animals, really, if they're not efficient, if they can make soy burgers that would be, much better than a hamburger, then when you went to the McDonald's, you'd order a soy burger. But the comparison here, though, is a hamburger you can get for the amount. This is the amount that USDA says that you're supposed to have in your diet every day, 50 grams of protein. So a hamburger you can get for $2. Soy burgers takes $3.45. So obviously the animal is still providing the nutrients at a reasonable price. So all these animal protein products are fairly competitive with everything except for dried lentils, and there's some dried beans that you can purchase, and peanut butter. That's about the only thing that's cheaper than some of these animal protein sources. Rice and bread, you have to eat 17 servings per day to get enough protein. So clearly that's not a reasonable option for most people. And mixed nuts, they taste good, but they're not as cheap as some of our animal protein sources. And another way to measure success is how many people are we really feeding. And these are statistics broken out by continent from the year 2000. Asia had 3.6 billion citizens and 1.1 billion acres of farmland. So the number of people they were feeding was 3.2 people per acre. Africa, about half the population density. Europe, about a third of the population density of Asia. And here in North America, we're at about 1/4 of the population density of Asia. So clearly, we're not feeding as many people, and that's partly why we can talk about having very high quality food if we have less people to feed. So South America is a little higher than us. Australia and New Zealand have 100 million acres of land and no citizens. I'm not sure what's going on down there. So to conclude, people try to improve the genetic merit of their animals in each generation. The modern selection tools can help us to make faster progress. We've improved animal production, but more recently, as we've added these other traits, we've also begun to try to improve the animal health from a genetic standpoint. The animal protein from these livestock products is very affordable. Last conclusion, North America has relatively few citizens. The reason I mention that is as we consider how the law affects animals, it'd be good to also consider how the law may affect other people around the world. And speaking of other people, I have a couple to thank here. Mel Tooker, Suzanne Hubbard, and Mark McGuire helped me to put this talk together. And some picture credits from where I borrowed the pictures from. These were actual live animals. Okay.
Well, Dr. Van Raden has given us breaking news about the disappearance of human life in Australia and New Zealand, and we'll let Al Gore know about that so he can change his slide presentation. I wanted to speak briefly to a point brought up by Peter Bennett, the leader of TIPS this morning, about what's going on in Pakistan. I'm really proud of the American Bar Association and of Peter's efforts with regard to raising the issue of what's going on in Pakistan, and I hope everybody here pays a lot of attention and supports the ABA and the TIPS section in that regard. And now for a change of pace, we've now heard from Dr. Van Raden about Justice, the bull. And we've also heard about mating practices in college dormitories, which I didn't think was on topic, but we kind of roll with things today. I'm very pleased to have Dr. Wall here today, Dr. Robert Wall. I had a chance to get to meet him last night, and whenever you meet anyone who's fascinating, you find that the fascinating aspect of their life doesn't come from nowhere. Years ago, he worked in southern New Mexico with regard to landing on the moon and getting the astronauts off the moon, and I can't even begin to comprehend all of his background. And then he happened to end up with a goat, and you can just imagine where the goat took him. Okay. Dr. Robert J. Wall would describe his work in genetic engineering as focusing on producing healthy transgenic animals by efficient means. In other words, he's an expert on animal cloning. Dr. Wall received his PhD from Cornell University and now works for the Bioscience and Biotechnology Laboratory at the U.S. Department of Agriculture. Got it right this time. He is credited with discovering a method to, and this is amazing to me, visualize pronuclei in embryos. This discovery led to the production of the first transgenic animals. Thanks to the work of Dr. Wall and his laboratory, genetically engineered cattle are now resistant to the mastitis infections. They have proved that a genetic switch can stop a virally induced cancer. Probably you can say virally induced cancer more easily than I can. His group has also proven that by utilizing certain construct sequences, the production of functional, that doesn't sound like a cow, that sounded worse. The production of functional transgenic animals. Is that a commentary on my introductory remarks? Okay. The production of functional transgenic animals can be doubled. I am so excited to meet Dr. Wall because, like many of you, I wanted to know if he would be willing to clone me.
I'm not going to answer that. Good morning. I'm very pleased to be here. I work just outside of Washington and never get a chance to hang around with a bunch of lawyers, so this is really a pleasure. So the job that I've taken on this morning is to have a little session on molecular biology 101, and then I'll talk about how we make transgenic animals and clones, and by the end of that, Barbara will recognize there is a difference between those two, though they can be the same. And then I'll give you a few examples of what's going on in the industry, though you're going to hear a lot more about that in a detailed way this afternoon. So some basic definitions. You all remember from high school that a gene is the basic unit of heredity. And I want to point out something to you. In the last talk, Paul talked about genes, but except for the horn gene, he didn't show you one gene because he doesn't need to know what genes are making those traits happen. This procedure, transgenic technology, requires us to know the gene intimately. So DNA is the molecule of nucleic acids that make up that gene, and DNA is the molecule that is formed into a chromosome. We get one pair from each of our parents, and the chromosomes are in the nucleus of the cell. Now this is important for our talk about cloning later. This is where all of the genetic information is in the cell, except there's some little squiggly guys around here called mitochondria that make the energy for the cell. They have 13 of their own genes. Just keep that in mind. So here's a chromosome, and if we unravel the chromosome and look at that double helix, that famous double helix, we can actually look at the genes. And this is major oversimplification, but it'll do for today. Genes have basically two parts. They have the coding region that usually gives the gene its name. The DNA sequence that encodes growth hormone would be right there. It also has a regulatory portion to the gene that turns the gene on and off. Your sex steroid genes don't turn on until you reach puberty. Your genes for taste are specifically in your tongue and not in your fingers. So it determines where in your body the gene is turned on as well. So the fact that we know those different parts allows us to manipulate those parts and make new combinations. So there's probably a legal definition that you know better than I do about what a transgenic animal is, but from where we come from, it's a gene that an animal didn't get from its parents. We intentionally put it in. And the intentional part of it is important in the definition, because otherwise we would all be considered transgenic animals, because if you've ever had a cold, that flu virus left some of its genetics behind in you. It's not too important, though, because it probably didn't influence the next generation, because it didn't leave its genetics in your eggs or in your sperm. So what's a clone? Well, Barbara Glenn tells us that they're just twins, and that's basically what they are. They're more or less identical copies made by asexual means. So there are actually two kinds of transgenic animals. There are the somatic cell transgenic animals and the germline transgenic animals. We're going to focus on the germline transgenic animals because when we make a transgenic animal, we only want to make it once, and we want its offspring then to have the trait that we introduced into that animal. So we make sure that the transgene is in the germ cells of the animal. Somatic cell transgenics would be what we would call people that had gene therapy. Or you may have a nucleic acid vaccine. It's a very popular idea that hasn't really come to fruition yet, to use a gene to inject into you to cause an immune response. You would make the protein that you would be responding to in a natural kind of way. But these somatic cell transgenics don't pass their new genetic information on to their offspring, whereas the germline transgenics do. So how do we make them? Well, we build a transgene We collect unfertilized or fertilized eggs. If we collect them unfertilized, we fertilize them in the laboratory. We introduce the transgene into the embryos. We transfer the embryos to a surrogate mother. And then the hard part of the experiment is we wait. And if you're working with a cow, you wait 9 months to find out whether the experiment worked or not. If you remember nothing else from this talk, you need to remember this. This technology has its power in the fact that we can take a gene from a bacteria or a copy of a gene from a bacteria and put it in a cow, or take one from a bird and put it in a pig. There are no species barriers, so that allows us to potentially give animals characteristics that they cannot receive through natural breeding. And what we usually do when we're building a transgene, let's say we want to produce a pharmaceutical in the milk of a cow. And let's say the pharmaceutical is a blood clotting factor. Well, here's the gene for that. It has its own regulatory element on it, which probably turns that gene on in the liver where we make a lot of our proteins. And we have a gene that normally functions in the mammary gland, maybe a milk gene. Well, we take the regulatory element from that milk gene, fuse it to the structural element of the clotting gene, and we would have a transgene, and that was what we would introduce into the animal. And that's what the operation looks like. This is actually a mouse egg with a needle that contains a solution of the genes being injected into a pronucleus of an egg. A pronucleus of an egg is a nuclear structure that contains either the genetic material from the sperm or the genetic material from the egg before they coalesce. So the microscope looks like that, and you could buy a Mercedes for what it costs for one of those things, and that's just a close-up. Oops. So cloned animals, on the other hand, don't necessarily have any new genes in them, though it turns out cloning is a wonderful technique to use when you're trying to make transgenic ruminants, at least. But they're nearly identical, and the nearly part is about those mitochondria. You'll see in a second how that happens. So we can make clones in several ways. The old way was to split embryos in half, and we could either do it at the 2-cell stage and have 2 embryos, or we could do it at the 8 to 32 cell stage. And this is one of the first sets of clones made in the early 80s by a company, Granada in Texas. And all these 7 animals came from a single embryo that was taken apart, and then the individual cells of that embryo were fused back into an egg. So if we've been making the clones since the early 80s, what's all the recent excitement about? Well, the recent excitement is about the fact that we're making somatic cell clones now as opposed to embryonic clones. And what does that mean, really? Well, or what's the implication of that? If you split an embryo, you're going to get twins that have whatever characteristics that embryo had, but you don't know what they are beforehand. But if you can use an adult cell and clone an animal, you can use it from an animal that you know the characteristics of beforehand, and that's where the power of somatic cell cloning comes in and why it's so important a technique. So how do we make clones? Well, we can build a transgene and put a transgene in if we want to, but that's optional. Otherwise, we collect unfertilized eggs, we collect cells from the animal that we want to make copies of, we make the cloned embryo, we do the same embryo transfer and wait. And here's sort of what it looks like. Irina will probably embellish on this this afternoon. The nuclear material of the egg is removed. Now, we have an oocyte without any DNA in it except for the mitochondrial DNA. And so that's still there. We take the cell that we want to make a copy of, and we have either put a gene in it or not. We fuse it to that enucleated egg, simulate fertilization so it starts to develop, and then the embryo, about 30% of the time in our lab, much higher in commercial labs, will develop to 8-day-old embryo that we transfer. So one of the advantages of cloning is that we can make genetic progress in a big hurry, or at least that's the promise. This is a graph of that same graph that Paul showed, basically, of a production trait, the value increasing over time. Well, if you have a stellar animal and you make a bunch of those animals, then your herd has a genetic potential that's lifted up immediately. Whether that's really true or not, the data is still coming in. So what do we use these animals for? Well, a lot of the transgenic animals in the 80s, when this process started, were used to build genetic disease models. We can now interfere with genes if we choose to do that, or add genes that overexpress something and mimic a human disease. And then that gives us an animal that we can study the disease, and otherwise we would just have to go around and look for mutants out there that may simulate a disease. There are a number of companies that are making nutraceuticals, food that has pharmaceutical-like activities, making pharmaceuticals, and you heard this morning about pigs being engineered to use for transplantation into humans. In agriculture, which is where we focus our attention, we're trying to improve food safety and animal well-being, as well as enhance production characteristics. And I'll tell you a little bit about one of those experiments at the end of the talk. So what's farming doing these days? Well, two examples of making pharmaceuticals in the milk of goats in this case are one, antithrombin 3, which has the distinction of being the first pharmaceutical made in a transgenic animal that's actually on the market. It was approved for sale in June of this year in Europe. The company that makes this is Genzyme, Transgenics. A company out of Annapolis is making butyrylcholinesterase in the milk of goats for the Defense Department. Butyrylcholinesterase is a nerve agent scavenger, so you get a shot of that when you've gotten exposed to nerve gas, and it takes care of the problem. DOD is paying for that, and I don't know exactly where that is in the regulatory schema of things. An example of a project being done in the Netherlands to try to improve the healthfulness of food is a copy of the lactoferrin gene from humans was introduced into a cow, and now these cows make human-like lactoferrin in the milk. That's a popular infant formula additive in Asia. It is not on the market yet, but this was actually the first bioreactor project conceived of, and the first one made in cows. So that's almost 20 years ago now that project started, and it's still not in the marketplace. So you can see that there's a fairly long timeline here. And I think it's sort of hard to deny the benefits of heart-healthy bacon. In this case, folks in the University of Missouri, in collaboration with some researchers at Yale, used a copy of a gene from C. elegans, a worm, to introduce into pigs that converts their omega-6 fatty acids into omega-3 fatty acids, the good fatty acid that's in fish. So let me finish my talk by telling you about a project that we're currently working on. It concerns trying to address this issue of mastitis. And as Paul said, it's an infection, a bacterial infection of the mammary gland. Every dairy in the world probably has some cases of mastitis in their herd. In the United States, it costs the dairy industry about $2 billion a year to address that issue. Partly it's in vet bills, but partly it's also in the fact that they have to throw the milk away when they're treating the animal, and because the quality of the milk is diminished when bacteria are growing in the mammary gland. Well, what we have chosen to do is use a copy of a gene from staph simulans that encodes lysostaphin, to introduce that into the cow so she expresses lysostaphin in her milk. And here's an agar plate with a lawn of Staph aureus growing on this plate. And you can see if we take store-bought lysostaphin and dilute it out and drip it on this plate, we can get these zones of killing. Well, this is milk from 3 transgenic animals, and you can see one of them does a pretty good job, almost better than the store-bought stuff, in killing the Staph aureus. The other 2 don't do as well, and that's one of the common features of transgenic animals. You make several transgenic animals, and each one of them will treat that transgene a little differently. Some will express the transgene more strongly than others. We have an idea of why that is, but we don't actually absolutely know. Well, it's one thing to kill some bugs on a plate, but the real deal is to try to infect the animals and see how they do. So we have been, over the last couple of years, been doing some challenge studies where we will actually infuse Staph aureus up the teat canal of 3 of the quarters and infuse saline in the 4th quarter as a control. And then we collect milk every 6 hours. And we analyze the milk to see if there are any live bugs growing in the milk, an indication of an infection, and whether the immune system in the cow is starting to crank up to address the problem. Well, if we find 2 samples in a row of milk that have live bacteria in it, we will immediately treat the cow to try to protect her, try to keep her from dying, actually. So here's the data from that experiment. We have now infected 22 control cows. That's an equivalent of 101 glands that we have infused. And 78 of them became infected. So even when we put bugs up the teat canal, not always does the gland get infected, but the controls, 78% of them did. In the transgenics, we've done 82 glands, just 9 ladies being done over and over again. Only 6% of them have become infected. And those 6% were some of the girls that are making very low levels of lysostafin. So we've now calculated that we know how much lysostafin needs to be in the milk to completely protect the cows. So this is just the beginning of what transgenic technology can do for you, and I thank you for your attention.
We have more than 15 minutes for questions, so there really will be time to get into some depth. So for those of you who haven't written questions out neatly and legibly, I ask that you do so. I did want to acknowledge, and Jill Mariani indicated this at the beginning of today's address, that this is an historic gathering. The American Bar Association has actually put this program on its homepage. That's just astonishing. I also wanted to admit the risk for those participating in this program today, both in terms of speakers and in terms of sponsors and co-sponsors. This was probably the most risky, I'm guessing the most risky program the ABA has done in a while, and there was courage all around for this meeting and this gathering to take place. And Bill Reppy, thank you so much for making Duke available to us. And I see questions are coming in. Do you want to just take one?
Yeah.
Dr. Wall?
There's a question here about labeling cuts of meat in the supermarket cloned, if it comes from a clone. Well, we can't yet have our meat in the supermarket, as I understand it, and so there isn't any labeling of cloned or not cloned cuts, and though they're better. You want to speak to the labeling issue? Are you going to speak to the labeling issue? There are better people in the audience to answer that question than I about whether labels are going to
Why don't
happen.
you make a point of addressing that when you're on? Would that be okay?
Okay.
Okay, thanks. Oh. You know what? You, come down. Can you give us a short answer to that question?
I can give you a short answer right from here, I think.
They're recording it for the students, so
All right.
you're very kind.
Hi, I'm Larissa Radenko. I'm the Senior Advisor for Biotechnology at the Center for Veterinary Medicine at the Food and Drug Administration. At the moment, there's a voluntary moratorium on the introduction of food, meat, and milk from clones and their progeny into the food supply. And so, as Bob has correctly said, there is no labeling on any food product from clones because there are no food products from clones in your supermarket right now. We are in the process of finalizing our risk assessment and other materials that may be issued regarding food from clones in the food supply. And until that time, I don't feel comfortable talking about where we are going to be on the labeling issue. But those of you who know the law know that the reasons that FDA, the authority that FDA has for requiring labels is whether or not there are any material differences in the food and whether or not there are any safety concerns. So I would just ask you to keep that in your mind and keep waiting for the risk assessment to go final. Thanks.
Thank you. Well, we're going to get her next time as an official speaker. Thank you.
Paul has 3 questions.
Okay. Paul, why don't you come up?
All right, and they're all very excellent questions. The first one regarding food costs, do government subsidies play a large role as to why animal protein is currently less expensive than some forms of vegetable protein. I mean, myself, my salary is paid completely by the taxpayers. So whether that's viewed as a subsidy, I think is probably debatable. It's research funded by taxpayers, I think is not generally considered subsidy. Most of the breeding companies out there, both plant and animal breeding, generally all private sector. Government subsidies, we did have a lot of those maybe in the 70s and 80s, but most of the subsidies have been phased out, and now the prices are really set at the world market price. If you notice, the price of milk went up recently. That's because China is buying a lot of powdered milk from us right now. So it's really, the price of milk is at the world price, and the other commodities are the same way. And then another question, which I actually got when I practiced this last week. Should my list of plant products have included tofu, which is fairly inexpensive and high in protein? Well, we have a vegetarian in my office, even though it's an animal office, who asked me exactly the same thing. I did add some of the other, changed my list slightly, and I went to the grocery store. I spent a half an hour trying to search for tofu, which I had not purchased before. I was unable to find it. I did some online research and found that the price was competitive, but I was not able to document it exactly with current statistics. So that's, but for vegetarians trying to balance their diet, they need to consider the source and the quality of the protein in their diet, and that's a reasonable question. And the next two are sort of very similar. Does modern breeding increase risk associated with a limited gene pool? Should modern breeding be complemented by small-scale preservation of antique breeds to maintain a diverse pool? And let me read the other one because it's very similar. Are short-term economic concerns expressed in breeding and other genetic material promulgation efforts increasing the risks inherent to genetic monoculture? How is this being addressed? So one of the first big projects that we did when I started my job is to calculate these inbreeding coefficients to show the farmers that they were getting too much of a good thing on some of these famous bulls. And they really have tried to scale back and use a wider variety. Now we're importing a lot of bulls from other countries to mix into our population, but that's still within one breed. Project introduced just in May is I completely rewrote our programs to process all breeds and crossbred animals together in the same genetic analysis, so that there are now options to be able to move the genes from one breed into another. That may help. But as far as preserving genes, I do sometimes feel guilty about that. If I make one population too successful, does that drive everything else into extinction? And we do have a laboratory at Colorado State University which does preserve animal genes, embryos, semen, from all of the rare breeds, any sort of unusual populations. They have in long-term nitrogen storage that they will be able to bring those back at any time. It's obviously better to maintain them in live status, though, so that otherwise just sitting in a nitrogen storage doesn't, is not a quick way of getting them back. But those, I think, were quite good questions.
Are there any? Well, since this has to do with cows, it could go to either one of you. Since the dairy industry needs females to produce milk, is there any incentive to genetically alter herds so that pregnancies produce more females or fewer males?
Well, there's an,
Yeah, so we're going to end up with too many one kind and not enough of the other?
well, there's an easier way of doing that, and I don't think you really have to worry, because if you use normal breeding strategy, you're going to get 50-50 on average. But Paul mentioned that farmers are now using sexed semen, and so they can actually choose a breeding unit that is 90-some% probability that it's going to produce a heifer calf or a bull calf at their choosing for, I think Paul said, $40 surcharge, is that right?
Right.
So you can do that now, and that would be the easiest approach to skewing the sex ratio.
Paul, could you address the economic merit a little bit that you're working on?
How we
Yeah,
come up with
yeah.
these,
Yeah, there's lots of challenging noises in this.
okay, the how do we balance all these different traits? The approach we've taken, there's been a lot of papers on should we put the animal's welfare directly, how much should we value that? And until now, we really, as of now, we have only really considered, is the animal creating a profit? So the farmers that own these animals, if the animal doesn't make a profit, they're just going to sell them anyway. So they sort of depend on me to tell them, will I make a profit with this animal? The welfare questions is sort of separate. That's I don't know how to breed a happier cow. We don't measure the size of the smile, but clearly animals do benefit by having better treatment, and the cows that can make a profit, their owner is going to invest a lot more in making sure they have good housing, good feed, all those kind of things. So the way we balance the traits is just to try to look up what is the price of milk, what is the price of feed, how much does it cost to get a veterinarian when there's a calving, a difficult calving, and the mastitis, what is the actual cost to treating mastitis? So we balance the genetic selection with the other tools that are available to treat those conditions. And that, it may not be optimal, but we do our best to get really the exact value in terms of dollars and cents that these traits express in profit.
Did you want to address that? Okay. There are many people in this room who've had a lot to do with the development of animal law, among them Joyce Tischler, who's seated in the front, David Faber, who's over there, and of course our host, Mr. Professor Ruppy of Duke. He's been there for a long time, and he now has the very challenging job of telling you how to get to lunch.
Lunch is in a different building. It's called the Law Annex. And for those of you who have been at Duke, it used to be known as Gross Chemical Building, Chemistry Building, but chemistry moved to something brand new. So we've got the first floor. It's over there across Tower View. And to get there, the best way is you want to get to the parking lot at the rear of the building, that way. And that's where free parking is tomorrow morning also. The lot that you drove into for free parking today goes over to the football fans tomorrow, but our students and staff are not going to be here, so there's going to be lots of free parking in the lot that we will walk through as we go to lunch. And there will be a student attending the gate there from about 8:15 to 11:15. Any event, you can reach the parking lot, turn right, cross the street to Gross Chemical Building, enter on the first floor on either side of the front. We have a wonderful buffet. We plan to have our speaker begin about an hour from now. We're not exactly certain, but that's about it. If you go down to the first floor at this end of the building, you go out the door there and you walk through a garden to the parking lot and turn right. If you go that way on the third floor, which we are on, or the second floor, you can get all the way to the back and go down the elevator, go down the stairs. We'll see you all at lunch. Isn't it wonderful to have a conference in which the morning session ends on time? See you soon.
About 10 years worth of cases. The reality is that in the last year, there haven't been anything particularly interesting or shocking or fascinating in the partnership area. I think, having said that it always is important. There have been a couple of cases that you'll see in there that are just in another round of bad fact cases, but nothing dramatic in the whole field. We still use partnerships a lot. I hope you do. Remember that in reality, the government has never had much success in dealing with adjustments on a going forward basis, on a gift basis. If you create your family partnership or your LLC and you're going to dispose of limited interest, non-voting interest by way of gift or sale, there's not very much law that gives the government much to hang its hat on. There are basically two cases. There's a case called Hackle that came out of Indiana where in Hackle the question was, are you going to get the annual exclusion for gifts of limited liability company interests? The limited liability company owned a tree farm that didn't produce any current income, wasn't going to produce any assets, any income for years and years and years because it's a tree farm. The limited liability agreement provided for an enormous number of restrictions on the ability of the limited liability company holders to do much with the interests. It appointed the grantor, the donor, dad in this instance, basically manager for life over the LLC. And the tax court in the Seventh Circuit said that we don't think you've given away a present interest here, so we don't think you're going to get an annual exclusion. Wouldn't have mattered if it had been an exemption gift, but for annual exclusion purposes, it matters. The other case that the government's had some luck in is a case called Senda, S-E-N-D-A. You remember Senda? There, the people formed the partnership on a particular day, put the assets in the partnership on the same day, and gave away the interest on the same day. All on the same day. And the government came in and said, we can't tell if you created a partnership and gave away the interest and then added assets. And remember, if you do that, no discount, because what you put in is not discounted. Or whether you put the assets in and gave away the units. In that case, remember, there were some transfers of other units a couple of weeks later, got a discount there. It was just the ones done on the same day. Well, what's the lesson there? Don't do it on the same day. That's not very complicated. Don't do it on the same day. I mean, and I'm amazed anyway. Who can do all that stuff on the same day? I mean, can you do it at the time you work in lunch and doing your timesheet and calling the client about a bill? Can you get all that stuff done the same day? I mean, there's no way we could do all that in the same day. It takes weeks, months, years. I keep copies of that case, by the way, whenever a client calls me and complains about being slow. I say, slow has benefits. Here's a copy of this case. The client fires me, but is better educated at least. Anyway, the point being the government has had very little success in this area. The success has come where you put assets in and you give away the vote and you retain these other interests. So it's not a situation where the partnership should be abandoned. Indeed, in many situations now, where we're kind of waiting to see what happens with the estate tax, we have partnerships that are created that are just standing by. And so, man, give you a simple example. What do? You got a 75-year-old widow wanders in, and she's got $5 million. And she's in good health. She might live to be 90. She's supposed to live to be 85. Might die tomorrow. She's got $5 million. What are you going to do there? We got a $2 million exemption now. Supposed to have a $3.5 million. Some people talk about a $5 million exemption. So what do you do? Well, from my point of view, it seems to me almost a no-brainer to say, well, leave $2 million out of a partnership, put $3 million in a partnership, give the vote to the kids, and she keeps the limited interest. She keeps the limited interest there. And she uses the limited partnership interest to make annual exclusion gifts in 2007 and 8 and 9. We tell the family that if you really want us to do more, then we'll do some kind of sale or some kind of gift or this or that. But if what you really want to do is kind of be low-key about it because you really think the exemption may go up, this isn't a bad deal. Is mom going to get a deduction for those partnership units? I mean, in other words, are we going to get a discount? Maybe, maybe not. If she dies soon, no. If she lives a while, probably we're going to get some. We're set up, if they call me someday and mom's had a stroke, I can do a sale, I'm ready to roll. I can do a gift, I can do a sale, I can do something like that. If the exemption goes up a bunch, you get a $10 million exemption, then she hadn't done anything complicated. She's given away some interest to the kids, maybe the grandkids, a trust, whatever. But it's kind of medium planning. And I always tell the clients, it's wrong. I know it's wrong. It will turn out to be the case that we should have been more aggressive or less aggressive. But I don't know which one we should be. So we're going to go right down the middle. It's basically buying an intermediate term bond, right? If you knew what interest rates were going to do, you would know what to do, but you don't know what to do, so you just kind of punt and you do it in the middle. Or as my wife likes to say, well, this milk might be bad. I don't want to drink it. Let's give it to the cat. Well, if the milk's bad, throw it out. If the milk's good, drink it. No, we'll give it to the cat. There you are. So from my point of view, that's a pretty good use of partnerships, and still is something to be thought about. Page 162, you'll see one of those issues that fascinates commentators, and you don't care one whit about it. That's my guess, and that is that the government has said if you have something like a GRAT or a charitable remainder trust where you, a grantor, is keeping an interest, we know when you die we've got to include something in your estate, but how much are we going to include in your estate and under what provision? So let's take a GRAT, for example. I create a GRAT and I put $1 million in, I'm going to get back $100,000 a year for 12 years, and I happen to die in year 8. What's supposed to be included in my estate? Well, you got two provisions, right? You got Section 2036 says if I give something away and I keep the income, it's going to be in my estate. I didn't quite keep the income. I kept an annuity. And we got Section 2039. It says if I have an annuity, it's supposed to be included in my estate. But this isn't really a commercial annuity, and if you read the section, it doesn't really fit. So what do we do? Well, since GRATs were invented, put in the law in 1990, we've all thought, basically, that 100% of a GRAT is going to be included in your estate if you die during the term. Ditto with a qualified personal residence trust, right? You tell your clients, we can't make the term too long because you might die and then it's not effective. You've got to live out the term. That's what we tell them. We didn't quite know which section it was, but we just didn't care. That was what we thought the rule was. Government now has issued proposed regulations. Proposed regulations say that we're not going to apply Section 2039, the annuity section. We're going to apply Section 2036. We're going to treat an annuity and all as if it's a retained income interest. That's what we're going to do. Fine. Does that make a lot of difference? Well, if you read the comments that have been submitted to the proposed regulations, you'll discover that maybe there are some instances where 97%, 98% of a trust might be included as opposed to 100%. As a practical matter, you don't care. What's more interesting is that the regulations were extraordinarily poorly written. I mean, I think you just have to say. For example, there was an example in the regulation dealing with a charitable remainder annuity trust, and the trust wouldn't qualify as a charitable remainder annuity trust on the terms as written. When you just played with the math, it just didn't work. And that's just sloppy. I don't mean that it's the end of the world, take somebody out back and shoot them, but it's sloppy that's what's happening. The regulations also don't cover all the stuff that really ought to be covered in terms of how you really do grants, et cetera. But again, I don't think that's going to matter a lot to you in the long run. A couple of things in the insurance area. I just want to mention to you, page 168, you'll see that the government has now enshrined in a revenue ruling that the insured can sell a policy to a grantor trust, and that will not be a transfer for value for Section 101 purposes, which is a good thing. So you've got an insurance trust, and you've got somebody who needs to move a policy from an old trust to a new trust. You've got more ways to do that now. You've got more ways to move an insurance policy out of somebody's estate without waiting the three-year period. A key aspect there is you've got to make sure that the trust is truly a grantor trust for income tax purposes. You don't want to have any problems with that, and you'll see a little discussion about that. You can also make a little cross note on page 67. There's a discussion of a field service advice that the government has issued that's very odd. It says that if you have the ability to use all of a trust's assets, all the income and all the corpus, to pay a life insurance premium, if the trustee has the ability to do that, even if there's no insurance in the trust, that trust will be a wholly grantor trust. That's an odd ruling because nobody's thought that was the government's position in a long, long time. It's in a field service advice. You don't quite know, not only is it not precedential, it's normally not reviewed at the higher echelons, and so maybe you worry about that more than you do other things. But it's one to keep an eye on. Page 170, yet another set of proposed regulations. These are in the area of estate administration expenses, attorney's fees, claims, that sort of thing. Section 2053. You probably thought that the area was working fine, right? You probably thought people die and their expenses, and you deduct them on the return. If you don't quite know what they are, you put an estimate, you tell the government how you got the estimate. They can audit or not audit, and off you go. Unbeknownst to you, there are enormous difficulties in this area, which the government has come here to help with. And they've come here to help with just a very odd proposal. These proposed regulations say that you don't get to deduct claims on an estate tax return until they're actually paid. Don't get to deduct them just because they might be going to be paid. Don't get to deduct them on what the appraised value would be on date of death. You get to deduct them when they are paid. So that's the example that comes up immediately that's caused people the most trouble is decedent dies in a lawsuit with somebody else. There are claims the decedent has against others, maybe a former employer. There are claims the former employer has against the decedent. Right? That's what you got. And so normally what we would do is we would say, on that basis, do we think that our claims are better than their claims? What do we think it might net out at the end? And we put a value there of either how much we think we're going to get or how much we think we're going to have to pay, and we're done. And there's uncertainty in all that, naturally, But there's uncertainty in lots of stuff. And you send it off to the government, and then the IRS can audit or not audit, and off you go. Pretty simple, straightforward kind of deal. You can't do that under these proposed regs. Under these proposed regs, claims you have against the employer are an asset of your estate. You value that at date of death value. And you put it on the return. So let's suppose that somebody says that you've got a claim, it's somewhere between 0 and $2 million, and you got a 50-50 chance of winning. Well, that's $1 million claim. Maybe there's a time value of money something, so it's not really $1 million, but let's call it $1 million. I'm supposed to put $1 million asset on my return. Let's suppose they got claims against me of 0 to $3 million with a 50-50 chance. So I really think at the end of the day, I'm going to have to pay these people $500,000. I don't get to deduct that until I actually pay it. So I have to pay a state tax on my claim, and I don't get this deduction until later on. And if you do it up in different situations, and if you look at the comments that have been submitted, you can see that it produces ridiculous results. And there are lots of other similar kind of circumstances. For instance, my favorite, you got a farm. Every farm in Kentucky is required to have a sinkhole in which you put fertilizer and oil and stuff like that. It's just required. You have to have that. It's in the back somewhere, covered by bushes, but you got it. And you get ready to sell the farm, and somebody says you got to do an environmental, et cetera, et cetera. Well, if I got $1 million farm and I have a cleanup cost of $50,000 to clean out my hole, but if somebody really tested, maybe I've got other environmental issues. Does that reduce the value of the property? Or is it a separate claim that I can't deduct unless I actually have to pay it? What's really going on there? So when as I say, when you read the comments, you discover things like that, and then you discover a really, really big gun. The point is made that this blows the marital deduction. How does it blow the marital deduction? Well, the marital deduction regs say you only get the marital deduction for things that you actually have that will go to the surviving spouse valued as of date of death. So the marital deduction requires a snapshot at date of death. I've got an estate, and let's suppose that I have this potential claim. It's from 0 to $3 million, and there's a 50-50 chance that I may have to pay. The marital deduction regs say I don't get a marital deduction for $1.5 million. 2053 proposed regs say you can't deduct that until you pay it. So that means I have an estate that has the simplest will in the whole world, all assets to spouse. I don't get an unlimited marital if you read these proposed regs. Obviously, an enormous defect in the system. So, if you don't like to read things that are proposed that are going to be changed, flip the page, because they're going to be changed, because they don't make any sense. If you like to rummage around in all that sort of stuff, then you can rummage around and you can kind of think about it. But it is it's an issue that I think you have to take a look at. Okay, let's skip along to page 209. 209. This is only an issue for you if you have a client with an old general power of appointment marital deduction trust. By old, I mean a marital deduction trust that was created before September 25th, 1985, and it's a general power of appointment marital. And the question that you have is, it's an irrevocable trust. Is it grandfathered for generation skipping tax purposes? The code says if it's irrevocable, September 25, 1985, it's grandfathered. It's an irrevocable trust. If you have a general power of appointment in somebody who's alive after September 25, 1985, stays alive today, so they can do whatever they want to do to the trust, is it really irrevocable? And that's the question. And interestingly enough, you've had a variety of court opinions on that. The first one was a case called Peterson in the early 1990s. Peterson was a Second Circuit case, and in Peterson, the court said, nope, the general power basically makes it not irrevocable. In about 1999 and 2000, 2001, you had a couple of cases called Simpson and Bachelder that came out, circuit court opinions, where they said, we don't understand what the issue here is. Whether it's a general power or not doesn't change the fact that it's an irrevocable trust. It was irrevocable. Nobody could revoke it. Go away. Go away. Government has changed its regulations in an attempt to thwart a little bit of the reasoning of those court opinions. And now along comes a case called Gerson. And Gerson raises the same issue. Gerson's going to be going before the Sixth Circuit, and it's going to be the first case dealing with the new regulations. We have lots of these buzzing around our office, and so we're very interested in the whole issue. Page 220. Qualified severances. What's a qualified severance? You remember when the generation skipping tax was invented in 1986 that it had a very harsh rule. And the harsh rule was that if you had a trust that was $1.5 million and you applied $1 million of generation skipping tax exemption to it, it was going to be in perpetuity 2/3 exempt and 1/3 non-exempt. So that in perpetuity, if you made a distribution of $1, if you gave it to the children, there was going to be some generation skipping tax exemption wasted, basically. If you gave it to the grandchildren, you're going to pay some tax. And there was just nothing you could do about it. Once you got a partial inclusion ratio, you were stuck. You were stuck. And it took a long time until Congress finally changed the statute and the IRS changed the regulations so that now you are allowed to divide that trust. And that's been a source of controversy about how can you divide it. Other people who can play games over the division, et cetera, et cetera. We now have final regulations on qualified severances. We have a couple of little amendments to some other regulations that are in proposed form. The nub of it is that they've tried hard to keep you from playing games, but you can do a division on what amounts to a fractional share basis. You can't do it on a pecuniary basis. In other words, in my example, $1 million and $1.5 million. I can't say that I'm going to put $1 million one place and everything else another place, but I can say that I'm going to put a fraction of the trust, the numerator of which is $1 million and the denominator of which is the value of the trust, one place, and the remaining piece another place. And the goal there that the government has is that you don't cook up this severance and wait for assets to go up or down and then actually do it. And I don't think that that's a big deal in the world, quite frankly. This is just a helpful thing for those situations when you accidentally could or did or somebody created funky inclusion ratios. So to me, it's a pretty good sort of result. Page 226. This is a ruling from the service, private ruling from the service, on the joint purchase of a residence. And I find that a lot of people have never done a joint purchase, so I put it in here because I want you to think about this. You know, you got a normal CUPR, right? I take a house, I stick it in a trust. I retain the right to live there for some period of time, 10 years, let's say. I retain a reversion. If I die during the 10 years, the house is back in my estate, fine. And I value my gift, subtracting my right to stay there 10 years, and my reversion. Fair enough. At the end of the 10-year period, normally the trust stays in a grantor trust, maybe for the benefit of my wife and my kids, and I pay rent to it. Fair enough. I make a gift, and the gift depends on the 75/20 rate at the time. Higher, the lower the gift is. How long the term is. Longer the term, lower the gift is. How old I am. Older I am, lower the gift is. Fair enough. Joint purchase gets you to the same result, except the kids actually pay for the remainder interest. So from an economic point of view, it can be not quite as good because I have retained the right to live in the house my entire life. I bought the life interest. My kids have bought the remainder interest. So there never comes a time when I'm making rent payments to the kids, which is a tax-free gift, essentially. So from that point of view, a joint purchase is not as good a deal. However, if I have used my exemption, a joint purchase enables essentially a Cupert-type operation without paying any gift tax, which is a really good thing. It's a really, really good thing. This technical advice is particularly helpful because it deals with a husband and wife who have bought the life interest together. And so when you run the math, it's very compelling. A couple of 60-year-olds, for example, can buy a life interest and the kids pay for the remainder interest. The purchase of the remainder interest will be under, say, 20% of the value of the property. So if you've already used your exemption, it's a really good deal, or used most of your exemption, it's a really good deal. The other is, if you're dealing with property someplace in Florida or New York, California, that's very, very expensive, sometimes you almost have to have a taxable gift in order to get the property out, which you don't want, just because the property is so expensive. Okay, just want to mention a couple other things, and we'll close here. You'll see on page 242 a couple of transactions of interest. I'm not going to go over them, but I want you to remember that the new way the government wants to sort of enforce the tax system is by saying to practitioners under Circular 230, we're responsible for not getting our clients anything that's too risky, too avant-garde. By saying to appraisers, you're responsible for not being too aggressive. And if we dawn on something here, we happen upon something that's too good to be true, even if we, the government, can't figure out quite why it's a problem, we're going to stick it on a list of transactions of interest and make you file a bunch of forms and make your clients file a bunch of forms if they get in the middle of this. And we're going to count on the fact that you don't want to do that to enforce the whole system. Finally, on page 254, there's a note about the new Form 706, which you undoubtedly have seen. It has a new box, and the box says, if a trust is in existence to which a grantor has sold stuff, check that box. Check that box. The idea here is to begin to track sales to grantor trust. I think that you can count on there being increasing audits in that area in the next few years, which is not a problem if you've been careful in doing your trusts. If you've been kind of cavalier on the grounds that, they're never reported, they're never seen, nobody knows anything about them, then I think you might want to go back and give a little bit of thought to how you're documenting those things. Y'all have been very attentive for early in the morning. I appreciate that very much. I think we're going to a break. Have a good rest of the conference. Thank you.